Today, we interviewed Dr. Eric Bombardier about his study that examines a protein known as sarcolipin in muscle regeneration. We chose to interview Dr. Bombardier because this study is one of the first of its kind at McMaster University to examine sarcolipin.
My name's Eric Bombardier, or Dr. Bombardier. I don't really go by doctor. I work for School of Interdisciplinary Science as an instructional assistant. I've worked in, I want to say, at least 7 or 8 different courses. So I've worked with pretty much every professor in this department. My hobbies, I like to fly fish in the summer. In the winter, really no hobbies. I stay inside.
How was your journey into medical research? Did you have any guidance that led you to research and this study?
My journey into research is pretty unique. I started my undergrad in '91, finished in '94, and then I did my master's in biology, looking at Atlantic salmon fish migration in Newfoundland. Finished that in '97. Then I went off and worked many different jobs. I did working, managing labs, teaching courses, etc. And that's basically how this cardiotoxin study came about. Was I was playing this a little more. Me and one of the students who's now a professor at Brock, we're in a car driving to see the Raptors play. During the car ride is where we came up with this idea of looking at the effects of sarcolipin on muscle regeneration.
Knowing your previous work on sarcolipin providing a novel muscle-based mechanism for adaptive thermogenesis, how did you come up with the decision to do this study?
Initially, the student I was talking about, Dr. Fajardo, was working on the role of sarcolipin in MDX mice, which is a muscular Duchenne dystrophy model, but in mice. And what we found was that in these mice, there was elevated levels of sarcolipin in the muscle. And that's because in Duchenne and muscular dystrophy is that the muscle it breaks down and regenerates continuously. So it's always continuously breaking down and regenerating. And then we saw that, hey, sarcolipin goes up in this model. Maybe it's because the muscle's regenerating continuously. And then on top of that, we found in previous literature where we found that during the embryonic stage of development, there was very high levels of sarcolipin in the muscle. Once again, showing that when muscles getting made, this protein is very high. So that led to the natural conclusion, or the logical pathway to what happens to sarcolipin during muscle regeneration after acute injury. And this is where we came up with cardiotoxin. And that's because cardiotoxin basically attacks muscle cells, and the heart is a muscle. But when it's injected directly into the muscle, what happens is it basically disintegrates all the myofibers, which is your muscle cells, but it doesn't influence or affect vasculature, nerve endings, any type of the nerves, any of the tendons, any of the ligaments. It only destroys the muscle, and it leaves the whole scaffolding still present.
What was memorable about this study?
Something memorable? Sure. There's a lot of memorable things about this study. We were the first to use it, pretty much, to look at cardiotoxin during the pilot studies that no one else had done on campus or anywhere else in any kinesiology department that I know of. So that was pretty neat.
How can the results of this study be applied to the treatment of musculoskeletal disorders?
Okay. This study is very much a basic study. So we are very much in the infancy of knowing how, whenever you talk about sarcolipin, you have to talk about calcium, because calcium is the most important signaling molecule in muscle. And it regulates pretty much everything. So the implementation of this in future medical research could have very much implications if we know that we can increase sarcolipin, and this will help with the regeneration of muscle. There are different ways that this can be done. For example, if an individual does have an injury, we know that cold acclimation will result in elevated sarcolipin levels in muscle. Two would be using CRISPR technology, right? Using CRISPR to augment sarcolipin expression in certain muscles, which could have very profound effects, especially in people with Duchenne's dystrophy.
What are the benefits of using animal models in medical treatment and therapy research? And is there a gold standard for animal models?
In cell culture first, bring it up to then maybe a mouse model, which the mouse model is great because you can either take sarcolipin out or overexpress it using genetic mouse models.
What do you see the future of these results being used for? What do you personally wish to see?
That's the next step, is figuring out how elevated sarcolipin levels in these mice influence the calcineurin and fat pathways, and then what does that do to which proteins are actually being produced.
Knowing the limitations of the study, what further research do you think can be conducted to better understand this topic?
It is injection site. We were injecting it into the tibialis anterior, which is a calf muscle, but we couldn't see it. So depending on where exactly you injected the cardiotoxin is where you're going to see the effect, right? So if you get a less than optimum injection, you're going to get less damage.
Do you have any future plans to do more research studies?
In sarcolipin and its effect on thermogenesis could be a potential treatment for obesity. Right? If we can increase sarcolipin levels in muscle of obese individuals, making those muscles less efficient, it would mean their basal metabolism would go up, thereby potentially helping to decrease obesity.
Last but not least, do you have any advice for students pursuing medical research and life sciences?
This piece of advice I could give is go volunteer in a lab. And it doesn't have to be a life science lab, it could be any lab, it could be a biochemistry lab, it could be a medical technology lab. That experience is something that you do not get just going to classes. Working in a lab, especially when you become a master's student, you learn, it's not really the content that you learn, it's more the skills.
Thank you for tuning in today and learning about sarcolipin and muscle regeneration, and a special thank you to Dr. Bombardier for giving us an inside scoop on his journey to produce this research.